All right, section 12.5 deals with the machinery of translation. We often associate, and rightfully so, ribosomes as being a very important component of translation, and that is absolutely 100% true. But we have to understand there are other components as well that make this complex process run. And so we're talking about various types of RNA. So not only messenger RNA, but we're going to see transfer RNAs, ribosomal RNAs that make up parts of that ribosome. There are other proteins called translation factors that are going to assist in the termination of the process and the identification of the initiating codon. And so this is one of the anabolic processes that really costs large amounts of energy to carry out. And so this is not cheap. We're going to use energy to proofread. We're going to use energy to establish translation complexes. There are various steps along the way in which we're going to burn through a triphosphate nucleotide in order to ensure the proper translation of the message. Let's start with transfer RNA. There are different tRNAs for the various amino acids that are going to be added to a growing protein. The example that they give here in your book is the tRNA for the amino acid serine carries serine at its 3' end. Now here's the important part of that. The fact that serine is carried on this tRNA means that the anticodon located here in one of the stem loops has to identify and bind to the codon that's specific for serine. And I think that's important as we go through. Understand there are two major areas that are important for the function of the tRNA. Not only the part that carries the amino acid, but more importantly the part that binds to the message and brings the proper amino acid to be placed in that specific position. So this is what's called the secondary structure of tRNA. Here's a three-dimensional structure. Notice even though RNA is single-stranded, it has the ability to fold onto itself through complementary base pairing using hydrogen bonds to stabilize the structure. And so we often call it a clover leaf in its two-dimensional structure, but you can see it sort of looks like a T here in its three-dimensional structure. Enzymes are very important here. The enzyme aminoacyl tRNA synthetase is in charge of adding the proper amino acid to its corresponding tRNA. So it's not as if tRNA is diffusing around the cell, amino acids are diffusing around the cell. They happen to run into each other and boom, an interaction is formed. There's an enzyme that's going to bring these together in their active site and do so very specifically. And so these enzymes number 20, one for each amino acid that's used to make proteins. And it's the ability then of these enzymes to add the correct amino acid to its tRNA that really becomes a second genetic code, because the specificity and the accuracy of this step is essential to everything that follows thereafter. Right? I mean, if this enzyme does not add the proper amino acid to the tRNA, translation is going to be incorrect. No matter how specific the anticodon is for its codon, if the improper amino acid's attached at the 3' end, the protein's going to be miscoded. And we know that miscoded proteins are going to have issues from a structural standpoint. We also know that they're going to have issues from a functional standpoint. So here's the enzyme drawn here with two distinct binding pockets. The first here you can see is a pocket to bind amino acids and its corresponding tRNA. But we also have a second binding pocket for ATP. We talked about the fact that this is energetically expensive. One of the ways in which we ensure that the proper amino acid ends up on the correct tRNA is through the hydrolysis of ATP. So what we can see here is that the amino acid and ATP bind in their respective binding pockets. Next, a tRNA diffuses in. Now, what I want you to understand here is that any tRNA can diffuse into the active site. It's whether we can establish productive interactions and, as a result, hydrolyze ATP. Will we get the charging of the tRNA? If the tRNA is not appropriate, those proper interactions will not be made, ATP will not be hydrolyzed, and that tRNA will eventually diffuse out. So this is the key step. The hydrolysis of ATP is the committed step, and that only occurs when the tRNA that binds into the active site makes those productive interactions. Once now ATP is hydrolyzed, AMP is released, causing the enzyme then to release the charged tRNA, which can then diffuse to the ribosome in order to assist in the translation process. This is a very important part of what we call quality control in the cell. A cell can't be 80% accurate. A cell has to be accurate as close to 100% of the time as possible. Of course, that doesn't mean it's perfect. It doesn't mean errors don't occur, but there are many steps that a cell takes in order to ensure the accuracy and the quality of the process. This is just one example. Now let's move on to ribosomes. Now for prokaryotes, they're one type of ribosome. It's the 70S ribosome. It's a little bit smaller than its eukaryotic counterpart. The main eukaryotic ribosome is an 80S ribosome. And so that's very important. We make the distinction. All cells have ribosomes, but not all ribosomes are created equally. Now, as I said, the main eukaryotic ribosome is 80S, but there are other versions of a ribosome compared to, depending on its cellular compartment. For example, the mitochondrion has its own set of ribosomes, and because that mitochondrion arose from a rudimentary prokaryotic cell, its ribosome is a bit different than the cytosolic ribosome that we're going to spend our time talking about. Now, ribosome can be broken up at multiple levels as far as the structure is concerned. At the first level, the most basic level, there are two parts to a ribosome. There's the large and the small subunit, based on the name, based on the size of each component. Here we go. Now, interestingly, this is not just a protein machine. You know, we've talked a lot about enzymes being made strictly of protein, but here's an example of a machine with catalytic components that are made of RNA as well, specifically ribosomal RNA. And these ribosomal RNAs play a very important role in determining the overall shape of that ribosome. Now here's the crystal structure of a bacterial ribosome. Notice here is the small subunit in blue. Here's the large subunit here in magenta. And within those sites are three distinct pockets called the E site, the P site, and the A site, each of which plays a very important role in the overall process of protein synthesis. Now it's very difficult for us to sort of see these pockets in this three-dimensional structure because it's static, right? We can't rotate it, we can't look from above, we can't look from the side. So in order to make this a bit easier, here's sort of the schematic model, an artist drawing, trying to recreate the ribosome. Again, here's the small subunit, 30S. Here's a large subunit, 50S. Here are the E, P, and A sites. We are going to learn the E site is called the exit site. Empty tRNAs, which have already donated their amino acids, exit through the E site. See, the thing that we have to recall from earlier lectures is that translation occurs in the 5' to 3' direction. So the ribosome, based on how this is drawn, is moving left to right. And so what we're going to see is the tRNAs are going to donate their amino acid. When the amino acid when the ribosomes move in this direction, what was in the A now shifts to the P. What was in the P is going to shift to the E site. And what was in the E site is going to exit. And so now let's talk a little bit about the P and the A. The P and the A is a peptidyl site. This is where we establish peptide bonds between the newly entered amino acid and the existing protein chain. So what we can see here is the polypeptide that's being built at this moment is present in the P site. The A site, the aminoacyl site, this is where we accept charged tRNAs that carry the correct anticodon for the next amino acid to be added to the chain. And so it's very important you orient yourself, at least have a basic understanding of what each site does, as in the next virtual lecture, we're going to break down each step in translation. Now, just as an aside here, we always try to focus on the genome and proteome aspects of how this applies to a broader picture. And it's very interesting. Ribosomal RNAs are very well conserved amongst individual species. And now we can use that conservation in order to establish relationships between different species. So, the translation machinery I talk about today didn't arise yesterday. It arose many, many years ago as we were establishing evolutionary relationships. So the original ancestor containing this translation machinery was the progenitor, which allowed us to establish all living things. You know, we talk constantly about the tree of life. You know, at some point there was an original cell that gave rise, or original organism that set up now the rest of the evolutionary tree. And so what we can start to do is look for the genes for these ribosomal RNAs within these genomes and look for differences as a way to establish evolutionary relationships. If two organisms have very closely identical ribosomal RNA sequences, you would argue that they branched off on the tree of life much more recently than two individual organisms that have major differences between their ribosomal RNA sequences. So here's an example. As we go down, humans, mice, rats, and then bacteria, E. coli, Serratia, and Bacillus. When you start to look at the 16S ribosomal RNA sequence here, notice that humans and mice have a lot of homology. I mean, if you walk through each individual base here, and I'm doing a quick scan, there's 100% homology. But yeah, now when you go to the bacterial species and you compare it back to humans, we can see there are major differences highlighted in red, suggesting then that humans and bacteria are not closely related, that when they branched off millions of years ago. And during the time of accumulating gene mutations, the ribosomal RNA genes diverged from one another. And so what we can now start to do, and we'll see this later on in the course in the second semester, is use similarities in gene sequences as a way to establish how closely two species are. And I think it's a very interesting approach.